Welcome. This is a review of a FIOS genomics report. The CCLE, or Cancer Cell Line Encyclopedia data from the Broad Institute of MIT and Harvard was analyzed in this report. This database contains many different data types collected using cell lines from different human cancers. In this report, we examine gene expression, copy number, mutation, and gene dependency scores, also known as sensitivity scores. This report includes a summary, metadata and quality control analysis, as well as association and functional analyses. Specifically, we looked at the BRAF gene, which has been studied extensively in cancer research. Notably, mutations in this gene occur in about 6% of all human cancers. We looked at data from 6 indications: skin, ovary, colorectal, bile duct, central nervous system, and lung. First, we looked at gene expression data and gene sensitivity scores across the cell lines. Gene expression data provides a snapshot of the activity of different genes across the cell lines. Gene sensitivity scores give an indication of how crucial a gene is to the survival of each cell line. For each indication, we grouped cell lines into high or low BRAF sensitivity groups and then looked at the relationship between the gene expression data and BRAF sensitivity by comparing the expression of cell lines with high BRAF sensitivity scores to those with low BRAF sensitivity scores. We performed statistical tests across the genes using linear modeling to determine significantly differentially expressed genes. In other words, we determined for each gene whether there was a significant difference between the gene expression levels in the BRAF sensitivity groups. In the skin cancer cell line indication, we found the most significantly differentially expressed genes between the high and low BRAF sensitivity groups. These results can be used to highlight potential biomarkers associated with BRAF sensitivity and begin to refine our understanding of the underlying biology and to help prioritize potentially novel therapeutic targets. We also looked at copy number variation data. This data indicates the genetic trait of the number of copies of each gene in each CCLE cell line. Analogous to the gene expression comparisons, we looked at the differences in the copy numbers between BRAF sensitivity groups for each indication using linear modeling. This analysis can highlight which genes show a significant difference in the number of copies of a gene in association with BRAF sensitivity. And finally, mutation data is a record of the differences in a cell line compared to the non-cancer equivalent. Here, we looked at the mutations between BRAF sensitivity groups and used Fisher's exact test to determine if there were non-random associations between the presence or absence of mutations and BRAF sensitivity. The results indicated that the presence or absence of mutations in the BRAF gene was significantly associated with BRAF sensitivity when comparing skin BRAF sensitivity groups. In conclusion, BRAF gene dependency in 340 CCLE cell lines was significantly associated with genetic features including variations in gene expression and copy number. The analysis indicated there were limited features commonly associated across 6 cancer indications tested. However, there was robust evidence that distinct genetic features could be utilized as biomarkers of BRAF sensitivity in each of the cancer groups. Thank you for watching this FIOS genomics report review.